Well, thank you, Dr. Bao and Subodh and Gator Bio for this opportunity to present today. So as Subodh mentioned, the title of the talk today is the development of a selective CD16A-based NK cell engager utilizing antibodies targeting a single amino acid variation. Again, this magnificent opportunity, really excited to speak on behalf of our company, Avantgen, on what we are doing, especially in the cancer immunotherapy and NK cell engager space. So I think we are all in agreement, and without a doubt, that immunotherapy, or harnessing one's own immune system to fight disease, has revolutionized the oncology field and the type of treatment and care we have today for patients. We have witnessed an explosion of the number of FDA-approved biologics in this area, and only within the past 10 years or so. First wave of drugs target novel checkpoint inhibitors, which allow your various cancer-suppressed white blood cells to fight back harder and stronger. The first one, ipilimumab, targets CTLA-4 and was approved in 2011, and was followed by multiple PD-1 and PD-L1 drugs. The most recent approval has been relatlimab, which targets LAG-3. All 9 of these are monoclonal antibodies, and they are being used to treat not only hematological malignancies initially, but have also been shown to be effective and safe in treating a variety of solid tumors, such as cancers in the skin, lung, liver, kidney, and gastrointestines. They are oftentimes being used as first-line therapy over chemo and in combination with other medicine. I mean, this advancement is truly mind-blowing. However, as these drugs' effectiveness are limited to a subset of patients, the need for new drugs is ever present, and we hope the next generation of drugs have further clinical benefit. The second wave in the last decade has been CAR-T, a type of cell therapy using engineered T cells, which usually have an antibody component to it displayed on the cell surface in order for it to bind to tumor cells and elicit its cytotoxic and helper immune functions. 6 of these therapies have been approved since 2017, so far all in hematological cancers. The third wave, T-cell engagers, are antibodies binding to at least 2 targets, one on the T-cell, usually utilizing the CD3 receptor, and the other to a tumor antigen, in order to redirect or engage the T-cell to attack cancer. And while blinatumomab was the first approved drug back in 2014, 3 more were very recently approved, with many more in clinical trials. With CAR-T and T-cell engagers, they have differences and currently have some limitations to overcome as well. In general, most of the current CAR-T therapies are autologous, meaning you need your own blood cells taken out, engineered, then put back in. On the other hand, an allogeneic or universal cell therapy would be more convenient or off the shelf to directly infuse any patient with pre-engineered T cells. In this aspect, pre-made proteins, such as antibody-based T-cell engagers, have a major advantage. And partly due to the above off-the-shelf factor, but also just the inherent biological complexity of cells versus a recombinant protein molecule as a drug, the cost of manufacturing and the logistics of treating a patient are significantly higher for CAR-T. One estimate of the total associated cost of CAR-T is about 5 times or more of a T-cell engager. On the flip side, CAR-T is usually a one-time administration and has a more durable response, as the cells can continue to grow in patients for up to years. T-cell engagers are usually administered by IV multiple times, as the drug is cleared from your system in hours or days. It is good to note that there are efforts to extend the half-life of these types of drugs. And probably what is more widely known are the potential adverse events of T-cell related therapy, like cytokine release syndrome and neurotoxicity observed in some treated patients. I want to make it clear that the FDA-approved drugs are deemed safe, and this illustration is a generalized and relative comparison. And lastly, both can be used relatively easy in combination with other drugs, which is great because there is a need to overcome some of these hurdles. So today, I'd like to explore with you an idea throughout this talk. What if drugs that recruit natural killer cells, or NK cell engagers, could overcome some of these challenges related to T-cell immunotherapies? NK cell engagers should theoretically have similar biologic drug advantages as T-cell engagers over cell therapy, but they would also harness another critical player of our immune system. What is an NK cell? In immunology, we were taught that an NK cell, shown here, is part of the innate immune system, the first rapid response to an infection. Meanwhile, the adaptive immunity relies on B cells and T cells to generate a tailored attack with so-called learning and memory characteristics. Adaptive immunity is why we get chickenpox only once and never again, and for the younger vaccinated generation who will never get chickenpox. And so antibodies and T cells have been all the rage in the field of medicine, largely overshadowing innate immunity, at least until recently. If we look at the lineage of an NK cell, it is something of a platypus. As opposed to all its myeloid-derived innate immunity partners shown on the left, the NK cell is a lymphocyte. But unlike T cells and B cells, NK cells lack receptors that undergo VDJ recombination, meaning they don't have receptors that can adapt to specific targets. Because they lack such special receptors, they were largely viewed as relatively homogeneous populations and comprising about 5 to 20% of the lymphocyte population. However, a study demonstrated that within any individual, we could have more than 30,000 phenotypically diverse NK cell subpopulations based on differentially expressed markers. Now, we are just beginning to understand that just like with T cells, there are functionally diverse NK cell subpopulations. We have also discovered that some of these NK cells have memory-like or adaptive traits, meaning they can persist and attack a subsequent infection of the same antigen months afterward. What makes NK cells unique compared to T cells and associated CAR T therapies is their cell-killing activity is HLA unrestricted, which has two major implications. One, NK cells can target cells that are not necessarily seen as foreign, but just abnormal, like tumor or infected cells. And two, it does not have the transplantation risk of graft versus host disease and related cytokine release syndrome, allowing for more suitable allogeneic cell therapy as CAR-NK, as an example. In order to select the right target expressed on NK cells for proper NK cell recruitment, we will need to first understand NK cells' various roles. There are a multitude of activating receptors, such as NKG2D and NKP46, and there are also a multitude of inhibitory receptors, including the killer immunoglobulin receptor family, or KIRs, as well as NKG2A. The total binding of activity and inhibitory receptors will tip the scales to whether or not the NK cell is activated. Once activated, NK cells can have multiple cytotoxic mechanisms against the tumor cell. One, direct lysis by secreting granules. Two, inducing FASL death receptor mediated apoptosis. And three, antibody-dependent cell cytotoxicity, or ADCC. Furthermore, NK cells secrete various cytokines that help recruit various other immune cells to eradicate tumor cells. CD16A, also known as Fc gamma receptor 3A, stands out as an attractive target as an engager for several reasons. While NK cells do not express receptors that can adapt to specific targets, as previously mentioned, CD16A binds to the constant domain of antibodies, specifically human IgG1 and 3 subclass isotypes, to elicit ADCC in a target-specific manner. By engineering bispecific antibodies with the CD16A engager and a tumor-associated antigen antibody, we essentially covalently join this pre-existing CD16A and antibody or two-part system into one and utilize the cell's existing natural machinery for all of its cytotoxic and helper functions. CD16A mediates both ADCC and induces cytokine production, such as interferon gamma, to recruit other cells. Also, it is important to note that the various receptors and cytokines illustrated are not all expressed constitutively and may change during the development or specialization of each NK cell. For example, NKG2A expression is lost upon NK activation. CD16A in particular is expressed on both activated and mature cells with enhanced cell killing abilities, and also on self-renewing adaptive NK cells, both types that are useful in targeting a tumor cell. Indeed, within the last 5 years, and this is not an exhaustive list, large biotech and pharmas have recently made high-dollar deals with smaller biotechs to develop NK cell engagers, and notably targeting CD16A. Developing antibodies targeting CD16A is compelling, both biologically and commercially. So how would we go about it? And what would be the ideal antibody profile? Well, we will need to first examine CD16A at the molecular level. CD16A is part of the type 1 Fc receptor family, with CD16B closest in homology. Each receptor has varying affinities to different immunoglobulin classes and have different effector functions. CD16A is expressed on NK cells, macrophages, and a subpopulation of T cells. And a single nucleotide polymorphism creates higher Fc binding and thus higher ADCC with the valine at position 176 than a phenylalanine. The allele frequency is roughly 40-60. And CD16B is expressed on granulocytes like neutrophils, which is the most common type of white blood cell. CD16B comes in two flavors, Na1 and Na2, and all of this is important to consider for the desired or target antibody profile, which is we want antibodies with high affinity for both CD16A, V, and F alleles. However, it should not bind to CD16B, Na1, or Na2 as much as possible. This is an important consideration for two reasons. One, neutrophils are the most common white blood cell and would act as a sink and absorb most of the circulating drug if it bound to CD16B. And two, neutrophils also have ADCC function, but use Fc gamma R2A as the receptor, while actually using CD16B as a negative regulator for ADCC, making it counterproductive. Functionally, it should block Fc receptor binding to other IgGs. This should be pretty easy to achieve as the interaction is weak. It's anywhere from the high nanomolar to low micromolar affinity range. And it should recruit and activate NK cells only in the presence of target tumor cells. For preclinical toxicity studies in non-human primates, we would also like to have cross-reactivity to the cynomolgus monkey CD16A. Now, when we align the amino acid sequences of the relevant target and non-target proteins, here lies the crux of the problem. The extracellular domains of CD16A and CD16B have a 98 to 99% sequence identity. In red are the positions where only one of the alleles, either on CD16A or CD16B, are the same, which is not ideal. And only three amino acid residues here shown in green are where they are the same in both alleles in CD16A and different in both CD16B, NA1, and NA2. Now, when we consider the appropriate methods for antibody discovery, we would argue that in this situation, any animal immunization-based discovery efforts would fail from the start, or at best might succeed, but with a less efficient and resource-intensive method. The first challenge of immunizing, let's say, a transgenic mouse would be to obtain a robust immune response, as FC receptors are highly conserved across species. If we are lucky enough to have an FCR knockout mouse on hand, or we break immunological tolerance and obtain a strong immune response, the second challenge would be to determine from serotiter the presence of CD16A specific antibodies, as the majority of antibodies raised will likely not be of interest and will mask any interpretable signal. Also, if CD16B is just slightly more immunoreactive than its counterpart, which we wouldn't know at this stage, it would make an already needle in a haystack type of intensive screening campaign even worse. So personally, I would not send our scientists down this hole. At Avantgen, our discovery platforms can efficiently tackle a wide range of targets, including difficult ones like CD16A. Our germliner library collection are very large yeast display libraries containing fully human antibodies that are displayed on the cell surface using a novel and proprietary technology independent from other companies. For each campaign, we introduce more than 100 billion yeast clones, and we have it in 3 antibody formats. We have our Avant-Epe, which is our single chain Fe library, our Avant-Foil or VHH heavy chain only library, and our Avant-Saber Fab formatted library, which is what we used for this NK cell engager program. In the second half of this presentation, we will demonstrate that the antibodies obtained with our discovery platform will meet the desired target antibody profile in expression, affinity, specificity, stability, immunogenicity, and other developability requirements. And for 3 critical and interdependent reasons: design, display, and screen. Design. We deep sequenced over 500 individuals to understand the antibody immune repertoire and formed a human antibody database. With this data, we gained insight on the general frequency of all amino acids at each CDR position for each antibody framework. The figure shows the distribution of a particular variable region heavy chain framework, where the germline amino acid is shown here underlined in red. And any other amino acids observed and its frequency are represented as the color-coded stacked bars. For example, for this particular variable region heavy chain framework, CDR2 position 55 has a glycine as germline, with a 40% frequency of having an arginine instead. Positions 56 through 58 are 2 isoleucines, followed by a proline, with no variation present. This is likely due to evolutionary and functional reasons. If we were to completely randomize or even partially randomize these positions, this could lead to problematic or immunogenic antibody sequences, as well as dilute the overall functionality of the library. So we don't. Another aspect of our design is that we use 100% germline framework regions, meaning no mutations in the regions between the CDRs, even if it was observed in our deep sequencing results. Framework mutations can naturally occur during somatic hypermutation in an individual, but that mutation could also be immunogenic in a different person, ultimately reducing an antibody drug's efficacy in a patient population. And lastly, we also removed most potential problematic sequence liability motifs, such as deamidation, oxidation, free cysteines, etc. By constructing semi-synthetic libraries with these general population weight distributions at each CDR position, and also allowing all combinatorial sequences for each chain with random pairing of CDRs and heavy and light chains, we find a practical way of introducing sufficient natural diversity with enough quote-unquote rearrangement to be able to obtain high affinity and developable antibodies without the need for further affinity maturation. So that was design. Display. A library is only good as how efficiently every antibody can be displayed on the cell surface. As a eukaryotic organism, yeast can properly fold and express proteins such as antibodies with similar post-translational modifications as mammals. Bacteriophage, on the other hand, has been shown to have unequal display across different frameworks, and there's also translational risk when eventually producing hits in HEC293 or CHO cells. Yeast also grows incredibly fast with a doubling time of about 90 minutes, allowing for rapid preparation of 100 billion clones for each discovery campaign. Screen. Utilizing successive rounds of magnetic sorting called MAX and fluorescence-based sorting, FACS, we can rapidly narrow down the pool of 100 billion clones to a panel of 20 to 100 functional antibodies in a matter of weeks or months. The quantitative sensitivity that comes from FACS allows us to enrich and identify the desirable clones efficiently. So we did that, just that with CD16A. We performed multiple enrichment sorts for the 176V and F binders, and a subtractive sort to remove the CD16B binders. We then selected yeast clones and induced secretion of Fabs to use for ELISA and flow cytometry analysis. A panel of antibodies were confirmed to have the desired specificity. The hits were then reformatted into IgG1 with a modified FC having silenced effector functions. The affinities of these antibodies to the relevant antigens were measured using the Gator instrument, BLI, and the results are summarized here. A panel of our antibodies had higher affinities to CD16A, F, V, and Cyno compared to a reference antibody currently in clinical trial, highlighted in blue. They also did not bind or bound weakly to CD16B, NA1, and NA2. In a cell binding assay with primary NK cells, A23 bound similarly as the reference. And with primary neutrophils, which express CD16B, our A23 and reference did not exhibit any binding to these cells, while our clone A21 only bound at the highest concentration tested. 3G8 antibody shown in purple here is a pan CD16 control antibody. With the Gator Bio system, we are also able to easily perform competitive assays to identify shared or unique antibody epitopes or bins. By first immobilizing CD16A onto the biosensor, then sequentially adding a pair of antibodies, we can determine if the second antibody is blocked or not by the first. In this figure shown here in the yellow line, 3G8 bound to CD16A in the presence of A23, meaning they have unique epitopes. The orange line shows A23 blocked itself and serves as a control and confirmed that the first antibody was bound to saturation. A summary of the results are shown here, where all of our antibodies share an epitope with the CD16A reference antibody, but not the pan CD16 3G8 antibody, as expected. In determining the epitope for our antibodies, we produced single amino acid substitution CD16A variants for 2 positions highlighted in green, the G147D and Y158H, to revert to CD16B in those positions. A CD16 control clone antibody was able to bind to wild type and these two variants equally by ELISA, confirming these proteins were intact. Clones A21 and A23 lost binding to the tyrosine 158H variant by over 100,000-fold, meaning that this single tyrosine at position 158 is the single and critical residue that confers these antibodies CD16A ultra specificity. Shown here is a summary and comparison of clone A23 and the reference antibody. Not only did our clone have higher affinity to CD16V, F alleles, and cyno, its binding was less attenuated in the presence of 10 mg per mil human IgG than reference antibody. This IgG concentration is approximately the physiological concentration of antibodies circulating within our bloodstream. And while the CD16A interaction with IgGs is rather weak, the reference antibody was still blocked by this level of IgG, and presumably the same attenuating effect could occur in vivo as well. Please take note, as we will observe this again, but even more pronounced in subsequent slides. Additionally, A23 outperformed the reference antibody in potential sequence liability, thermostability, and expression. The melting temperatures and aggregation temperature associated with our antibodies are consistently higher, meaning more stable antibodies that lead to improved expression, efficacy, and shelf life. Now that we have measured the biophysical properties of our CD16A antibodies, we then produced and tested them as bispecific NK cell engagers. Specifically, we coupled them with an anti-CD19 antibody in either a tandem scFv format or a bivalent tandem scFv fused with the IgG4 Fc. Using the latter bispecific engager format and a CD16AV reporter cell line, A21 and A23 are slightly more potent than reference in activating the engineered effector cell in the presence of CD19 expressing Raji cells. All three also activate more than another control shown in purple, which is the same CD19 binder, but as a monoclonal antibody with an enhanced ADCC Fc. To put this into context, the enhanced ADCC Fc used is the same as margetuximab, which is essentially a HER2 FDA-approved trastuzumab antibody with the modified Fc that demonstrated enhanced NK cell activation, ADCC, and improved clinical benefit over trastuzumab, and gained its own FDA approval in 2020. The fact that we see significantly higher activation levels with our NK cell engagers at the same concentrations is really exciting. As a control, incubation without Raji target cells, activity is not detected with any of the tested antibodies, demonstrating the requirement of having both CD16A and CD19 scFvs binding to their respective targets for activation. And strikingly, A21 and A23 retain their potency even in the presence of the 10 mg per mil IgG, while the reference antibody almost completely loses activity, shown here as the green line. In summary of the EC50 values is shown here, with our panel of antibodies having several orders of magnitude higher potency and activation compared to reference. In the same reporter assay, but in a monovalent tandem scFv format, we also observe potent Raji cell-dependent reporter cell activation with A21 and A23. Reference antibody in this monovalent format has significantly lower potency as well as lower max activation, both without and with 10 mg per mil human IgG, again shown as the green line. Summary of the EC50 values are shown here. We next looked at functional ADCC-dependent tumor cell killing using primary NK cells co-incubated with our antibodies and Raji cells. In 2 separate donors, our antibodies mediated higher maximal NK cell-dependent Raji cell killing. And in the presence of 10 mg per mil IgG, A23 retained activity while the reference did not. And in the tandem SEFV format, we are observing around 1 order of magnitude higher potency with our A21 and A23 antibodies compared to reference. In conclusion, direct comparison with the leading reference CD16A clinical antibody demonstrated Avantgen's clones outperform in terms of both developability and engager activity across various formats and assays. So, can NK cell engagers overcome the challenges of T-cell immunotherapies? In the near future, we will see how some of the first generation NK cell engagers will perform in phase 1 and 2 trials. And we believe the results will be quite promising, yet we'll probably need further improvement. Our next gen NK cell engagers and CD16A antibodies, coupled with your own novel target antibodies, are available for licensing and co-development. If you're interested to learn more, just check out our website at avantgen.com, reach out and become a partner. Thank you so much for your time and attention. Happy to take questions. Thank you.
Dr. Wong, for you, and I'm paraphrasing it, you went over CAR T and T-cell engagers and NK cell engagers. What are your thoughts on CAR NK?
That's a great question. Yes. So obviously there's a gap where I did not mention CAR NK just for sake of timing. But if for those that don't know, CAR NK is a cell therapy just like CAR T, but instead of using engineered T cells, you're using the natural killer cells. And in fact, there's actually more cell therapies with CAR NK going on in the clinical trial as we speak. And the NK cell engagers is probably a step later than half a step later than that. When I think of CAR NK, there's definitely similarities to the CAR T in terms of both the advantages and the limitations, right? It is a cell therapy drug, so you're still going to get, even if it's allogeneic and universal, you're still going to get significant costs in manufacturing and complexity compared to a single recombinant protein, right? Such as an NK cell engager. And then the other aspect is that CAR NK, I believe, will be deemed safer than CAR T, generally speaking. But again, if you don't get a complete cure from that initial cell therapy, what's very likely to happen is that you're going to have to have a follow-up treatment with either a let's say anti-PD-L1 medicine or other recombinant protein antibodies, and very likely to include NK cell engager as an example. Because if we look at CAR T therapy and what's going on there, oftentimes they'll get successive treatments with a T cell engager later, because it's kind of hard to get continuous treatment of cell therapy. But with with the recombinant protein, it's a lot more manageable than that. And the other thing is that the one part of CAR NK or CAR T is that it would be pretty challenging to have a combination therapy where it's 2 different cells. So again, speaking to the importance of having these engager molecules, T cell engagers and NK cell engagers, they're more amenable to combination therapy. Have a good day.